Welcome to part 21 of this video on the immune system. Okay, so we're currently in the process of discussing B cell production in the bone marrow, and the stage that we've got to so far is that the developing B cells, they have designed their own immunoglobulin heavy chain. So in particular, they've designed the variable region of their immunoglobulin heavy chain. They've now started producing this unique design of immunoglobulin heavy chain, and they've produced it in the membrane-bound IgM or IgD type. Now what we need to do is design our own immunoglobulin light chain, and of course that means designing the variable region of the immunoglobulin light chain. We then need to produce this immunoglobulin light chain, and then of course assemble full immunoglobulin molecules, which will be of the IgM or IgD membrane-bound type. So that's what we're going to study in this next video, the designing of the immunoglobulin light chain. Okay, so you might be thinking coming into this video that this process is going to hopefully be extremely simple, and it is going to be extremely simple. However, there is one complicating factor, and it's that one complicating factor that is going to haunt us for this video, and which we're going to have to try and overcome and understand. Okay, so let me introduce you to the complicating factor right away. So the complicating factor is that we do not just have one type of immunoglobulin light chain locus. So I would love to tell you that there is one type of immunoglobulin light chain locus. This is the chromosome it's on. Of course, we'll have two copies of it overall on the two homologous chromosomes, but other than that, it's exactly like the immunoglobulin heavy chain locus, except it's much simpler because we don't have all of those different constant gene segments which made the immunoglobulin heavy chain locus so complicated. That's not the case, however. Instead, we have two different types of immunoglobulin light chain locus. So we have what is known as the immunoglobulin Kappa light chain locus, which is present on chromosome 2. So of course you have two chromosome 2s, and therefore you'll have two overall of these Kappa immunoglobulin light chain loci. But then we also have another type of immunoglobulin light chain locus, which is known as the Lambda immunoglobulin light chain locus, and of course these are just Greek letters. So Kappa, as the actual Greek letter, looks like a tiny little K, and Lambda is another Greek letter, which looks like this. We also have a Lambda immunoglobulin light chain locus, which is present on chromosome 22. So of course, again, you have two chromosome 22s, and therefore you'll have two Lambda immunoglobulin light chain loci. So this makes it more complicated because now overall we actually have four different immunoglobulin light chain loci. Now, before I explain the significance of this, or rather the lack of significance of this, what I just want to do is explain what's going to happen. I just want to describe initially how our developing B cell is actually going to go through the process of designing an immunoglobulin light chain. And then at the end I will come back and discuss the significance, or rather, as I say, the lack of significance of this. So for the while, just go with it, and at the end I will come back and explain everything. But I think it's easier to explain the lack of significance of this once I've actually described what's going to happen. So that's how I'm going to proceed. So let's say then that I am one of these developing B cells. I have produced an immunoglobulin heavy chain, and I'm now turning my attention to producing an immunoglobulin light chain so that I can assemble full immunoglobulin molecules. Where does my attention go first? Well, I have four immunoglobulin light chain loci that I could use to make an immunoglobulin light chain locus. To make an immunoglobulin light chain. So which one am I going to go to first? Well, the answer is that they always go for one of the Kappa immunoglobulin light chain loci first. The Lambda immunoglobulin light chain loci are the ones that they go to last. So we're going to start off then by going to one of these Kappa immunoglobulin light chain locuses. And this is where it gets really simple now. So what I'm going to show you is what does the kappa immunoglobulin light chain locus look like in its germline form, i.e. the form that it will currently be in this developing B cell that has not yet begun the process of trying to design an immunoglobulin light chain. So, and then of course we'll go on to VDJ recombination for this immunoglobulin light chain locus to produce a final immunoglobulin light chain gene to produce an immunoglobulin light chain from. So, once again, let's let the two parallel lines here represent a piece of double-stranded DNA, and I'm going to show the different gene segments on the immunoglobulin light chain kappa locus as just boxes around the little sections of this DNA. So, we're going to have lots of variable gene segments and lots of joining gene segments in the germline form of the immunoglobulin light chain kappa locus. However, we're not going to have any diversity gene segments. So again, it was the immunoglobulin heavy chain locus that is the classical example of VDJ recombination because it has variable gene segments, diversity gene segments, and joining gene segments. Now that we're on the immunoglobulin light chain kappa locus, we've lost the diversity gene segments and we just go back to having variable gene segments and joining gene segments. So let me show this. So here, this is representing my section with lots of variable gene segments here, all spaced by introns as usual, and again, I don't know, neither do I think it's particularly important what the specific number of variable gene segments that are in this immunoglobulin light chain kappa locus actually is. So here, these in red are representing the variable gene segments of the immunoglobulin light chain kappa locus. So I'll just underline this. So this is our variable gene segment section. Next, of course, we're going to have our joining gene segment section. So I'll put these in here. So here's our first joining gene segment, our second joining gene segment, and it will go on all the way up to our last joining gene segment. So here is our cluster of joining gene segments, and we'll have those joining gene segments coloured in green here. So there's the first one, the second one, and then dot 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 all the way up to our last one, and again they're all spaced apart by introns there. So we have a whole selection of variable gene segments to choose from, and we have a whole selection of joining gene segments to choose from. The concept is exactly the same, that to make the variable region of an immunoglobulin light chain, all you need is one of these variable gene segments and one of these joining gene segments, stick them together, and that piece of DNA will now code for a variable region of an immunoglobulin light chain locus. And now it's really simple after that, because of course we now need the gene segment for the constant region of the immunoglobulin light chain, and unlike the incredibly complicated array of constant gene segments that we had for the immunoglobulin heavy chain, we now just have one constant gene segment here, so we just call that the C gene segment. So I'll colour this in orange. So that is incredibly simple, so lovely, beautiful. And now of course you know what's going to happen now. So this is the germline form of the immunoglobulin light chain locus, kappa. So what's now going to occur is VDJ recombination. So we will select a variable gene segment to use, we'll select a joining gene segment to use, we will cut out all of the DNA in between, and then we won't bother, again, remember, we won't bother to cut out the DNA between the selected joining gene segment and the constant gene segment. That's one of the things that it would seem intuitive to do, but one of the things that nature does not do as part of VDJ recombination. Instead, it cuts all of those remaining joining gene segments out at the level of transcription. So the pattern remains the same overall for VDJ recombination. So again, I'll just show this. So let's say again, I select as my chosen variable gene segment the second variable gene segment here, and I select as my chosen joining gene segment the second joining gene segment here. What I will now do is I will cut somewhere here, and again the same principle applies as always. The places where these cuts are performed are not set in stone, which will give rise to junctional diversity. And then we'll cut here, and we'll cut all of that DNA out and bin that DNA, and now we will join our chosen variable gene segment to our chosen joining gene segment, and this is what we will end up with after VDJ recombination is complete. So here is the first variable gene segment, which is the one that's in front of our chosen variable gene segment. So of course I chose the second variable gene segment, but if I chose one further along, all of the variable gene segments that were upstream of that would still be upstream of it. So here is first variable gene segment here in red. Here's our chosen variable gene segment. Then what we'll have is our chosen joining gene segment now connected to our chosen variable gene segment. So here it is in green. And then we continue on. So all of the all of the joining gene segments that were downstream of the chosen joining gene segment, they are still here. So dot dot dot, and there's our final joining gene segment in the array of joining gene segments. And then here will be the constant gene segment here in orange. So this is what we've got after VDJ recombination has taken place on this immunoglobulin Kappa light chain locus here on chromosome 2. Okay, right. So again, I'll stress that the two different ways that we're producing diversity here are combinatorial diversity and also junctional diversity. So combinatorial diversity because we've got a huge number of variable gene segments and a huge number of joining gene segments that we could choose. So there's a vast number of combinations that you could make there, and they will all produce a slightly different portion of DNA that's actually going to code for the variable region of our immunoglobulin light chain. The junctional diversity comes from the fact that there is a variable amount of DNA in between these two chosen gene segments here, and that will lead to further variation in this portion of DNA that's going to code for our variable region of the immunoglobulin light chain. And that's how all of these different developing B cells are going to design a different immunoglobulin light chain to actually produce. Okay, of course the cost of this is that it can go incredibly wrong, because we can end up reading the joining gene segment out of frame, and then it's just nonsense, and then what we produce won't actually function as an immunoglobulin light chain. In that case, what will happen is the cell will then have to go onto its Kappa immunoglobulin light chain locus on the homologous chromosome 2. So the first move is to go to the other Kappa immunoglobulin light chain locus on the homologous chromosome. Then if that fails as well, so if you get both of the Kappa immunoglobulin light chain loci failing to produce a viable immunoglobulin light chain locus, you can then go onto the Lambda loci, and the Lambda loci have a very similar structure and a very similar process of VDJ recombination to design them. So that's the order in which these developing B cells will go through the different immunoglobulin light chain loci. They'll go through both Kappas first, they'll try both Kappas, and only if the two Kappa immunoglobulin light chain loci fail, then will they go onto the Lambda immunoglobulin light chain loci. Okay, so we'll come back to discussing the difference between the Kappa and Lambda immunoglobulin light chain in just a moment. Let's just complete this story up here, because what's now going to happen is our VDJ recombined Kappa immunoglobulin light chain locus, presuming that it has done well, so let's say that this VDJ recombination of this Kappa immunoglobulin light chain locus was successful. What will now happen is transcription will take place, and you'll start transcription from your chosen variable gene segment here, and you'll go all the way down to the end of the constant gene segment here. So this is the piece of pre-mRNA that you'll actually produce here. So you'll have your chosen variable gene segment there in red, you'll have your chosen joining gene segment there in green, then you'll have all the other joining gene segments, so dot dot dot, up to the final gene segment here, then an intron, and then of course we have the portion of mRNA that pertains to the constant gene segment here. So this is our pre-mRNA. Now what we have to do is just cut out all of those introns and all of those joining gene segments that we're not going to actually use. So splicing will now cut all of this out here. So this will be removed by splicing. And then the mature piece of mRNA that we'll get will look now like this. So we'll have our chosen variable gene segment here in red, we'll have our chosen joining gene segment here in green, and we'll have our chosen constant gene segment here in orange. So this is now the mature mRNA, and this will code for our immunoglobulin light chain. So the variable and joining gene segment, they'll code for the variable portion of the immunoglobulin light chain, and the constant gene segment, that will code for the constant region of the immunoglobulin light chain. Right, so that's the way in which we build an immunoglobulin light chain then. The way in which we design and then build an immunoglobulin light chain. So what I now want to discuss is the significance of these different immunoglobulin light chain loci, the Kappa immunoglobulin light chain loci and the Lambda immunoglobulin light chain loci. And by the way, the Lambda immunoglobulin light chain locus, it will go through a similar process of VDJ recombination as we've described here, and I'm not going to go through that separately now. Okay, the big difference is that if you produce an immunoglobulin light chain overall from a Kappa immunoglobulin light chain locus, then the constant region of that immunoglobulin light chain that you overall now produce will be different to if your immunoglobulin light chain is produced by a Lambda immunoglobulin light chain locus. So there are different types of immunoglobulin light chain which will have different constant regions basically, and there are two different types. You can either be a Kappa immunoglobulin light chain and then the constant region of that immunoglobulin light chain is of the Kappa type, or you can be a Lambda immunoglobulin light chain and then the constant region of that immunoglobulin light chain will be of the Lambda type. So just drawing this out here, here's a picture of an immunoglobulin light chain. So remember, it has these two important immunoglobulin domains here, like so. So here is the variable light domain, and here is the constant domain of the light chain, like so. Now the variable immunoglobulin domain here is the one that we're designing, so in both cases that will be designed, but now we're saying that for this constant immunoglobulin domain of the light chain, there are two options. We could either have it of the Kappa form, or we could have it of the Lambda form. And what I now want to ask is, what's the significance of that? Does it have any significance? Well, firstly, note the difference between this and the different options for the constant region of the immunoglobulin heavy chain. In the case of the constant region of the immunoglobulin heavy chain, all of those different options, so those 10 different options that we saw, the reason you had those 10 different options was because you had those 10 different gene segments in the immunoglobulin heavy chain locus. Here, the reason that you have these two different options for the constant region of the immunoglobulin light chain is very different. It's because you have more than one type of immunoglobulin light chain locus, so there's a different reason. The second thing to say is that once the designing process of the immunoglobulin light chain locus is completed during B-cell development, then it is absolutely set in stone what that developing B-cell will use as its constant region for its immunoglobulin light chain, because either it will have designed its immunoglobulin light chain using a Kappa immunoglobulin light chain locus, or it will have designed it using a Lambda immunoglobulin light chain locus. And after it's completed that design process, it will be set in stone. It can't suddenly then decide, actually we'll use the constant gene segment of the other type of immunoglobulin light chain to make our immunoglobulin light chain. It's set in stone after the development process is complete. So each of these B cells that will be coming out of this development process, it will only be able to produce an immunoglobulin light chain of either the Kappa type or the Lambda type. Whereas for the immunoglobulin heavy chain, you will remember that we haven't actually done anything to that region of the immunoglobulin heavy chain locus where all of the different constant gene segments are. They have been left well alone. So if I just bring back up the picture, here is the picture of our immunoglobulin heavy chain locus. As far as VDJ recombination was concerned, we did not alter this in the slightest. All of that is still here. I know I didn't draw it all out again, but it's all still here. So overall, the B cell that is coming out of the production process is still actually capable of producing immunoglobulin heavy chains where we use any one of these 10 constant gene segments to code for the constant region. So it still has the possibility of making immunoglobulin molecules, immunoglobulin heavy chains rather, of those 10 different types, whereas it's not the same for the light chain. Now, at the moment I've said that it's only producing immunoglobulin heavy chains of the IgD or IgM type, but that's because it's only actually transcribing as far as the constant delta region. It still does have the possibility that it could do some tweaking around here and suddenly start producing immunoglobulin heavy chains of other types, and we will see how it can do that later on. So, the big difference then is that the decision of which constant region you are using for your immunoglobulin light chain is made during development, and it cannot be changed now. So once design of the immunoglobulin light chain is finished, the constant region of that immunoglobulin light chain is set as either Kappa or Lambda, and of course the variable region is also set after we've designed it. Okay, whereas in the case of the immunoglobulin heavy chain, the constant region can still be tweaked. We can still change which constant region type of immunoglobulin heavy chain we're actually going to produce, and we will see how that is actually going to occur later on. So, that's the difference between this and the immunoglobulin heavy chain. The second thing that I want to discuss is what is the significance of this? Is there any significance of this? And the answer is we don't think that there is any significance of this. We do not think that it makes any difference to the function of the immunoglobulin molecule, either membrane-bound or secreted, whether the immunoglobulin light chain is a Kappa immunoglobulin light chain or if it's a Lambda immunoglobulin light chain. It does not seem to matter at all. So unlike how I've said that the choice of constant region for the immunoglobulin heavy chain does change the function of the immunoglobulin molecule, it does not seem that whether you have a Kappa immunoglobulin light chain immunoglobulin light chain constant region or a Lambda immunoglobulin light chain constant region, it doesn't seem to make any difference to the function of the immunoglobulin molecule. So overall, this does not seem to matter. So all you should think about with regards to these two different types of immunoglobulin light chain is that they are there as a backup. The initial starting position is that you want to produce an immunoglobulin light chain of the Kappa type, and you will use both of the immunoglobulin light chain Kappa loci first, and if both of those fail in the VDJ recombination process, then you can move on to using this immunoglobulin Lambda locus. And just be aware then that the immunoglobulin light chains that you produce if you use the Lambda locus will have a slightly different constant region to those immunoglobulin light chains that you would have produced if you had used a Kappa immunoglobulin light chain locus. Okay, right, so that is the design of the immunoglobulin light chain now complete. So our developing B cell will go through this process, and once it has designed its immunoglobulin light chain, it will produce those copies of this immunoglobulin light chain, and these will assemble with the immunoglobulin heavy chains to produce a full immunoglobulin molecule. So now, the position that we will actually be in is we will now have B cells in the bone marrow. So let me just actually draw this process in. So here is the lymphoid progenitor cell. So this can be the nucleus of the lymphoid progenitor cell. So I'll just colour this in. And I think I was previously showing lymphoid progenitor cells in orange. So this is a lymphoid progenitor cell. What it's now going to develop into is a B cell, like so. So here we go. This is called a B cell. And these B cells, they will have a design of immunoglobulin heavy chain, they'll have a design of immunoglobulin light chain, and they're going to produce both of these. So let me now show this. And they will assemble immunoglobulin molecules by joining the immunoglobulin heavy chains to the immunoglobulin light chains. And these immunoglobulin molecules will be of the form IgM or IgD, and they'll be membrane bound. So let me now actually show this. So here we go. On the surface of the B cell now, we're going to have our completed immunoglobulin molecules like so. So I'll just put in the disulfide bonds. So this is now what we would call a B cell receptor, and it is an IgM or IgD type immunoglobulin molecule, and it's membrane bound. So the heavy chains, they have the special bit that implants them into the membrane. So they're membrane bound IgM or IgD molecules, and they have the specific variable region that this B cell has now designed. Well, the lymphoid progenitor cell that became this B cell designed. So we'll have this B cell shown in red here. So this B cell will absolutely coat itself in B cell receptors, which will be IgM and IgD molecules that are membrane bound. And I want to stress it will have a mixture of the two different types. So they'll be coated in IgM and IgD membrane bound immunoglobulins, and these are what we now call the B cell receptors. So this is now a B cell in our bone marrow. What will now follow is a process of central tolerance. So the next process that will occur is central tolerance. Just like we did for the T cells, we have to make sure that these B cells that we're producing are not going to be directed against self-peptides. So what will happen inside the bone marrow is we will test all of the B cells that we're now producing with their new immunoglobulin molecules on their surface and make sure that the antigen binding region of these B cell receptors cannot bind to a self-peptide. And if they can bind to a self-peptide, the B cell will be destroyed. Now, this central tolerance process that occurs in the bone marrow is not as well understood as the central tolerance process that occurred in the thymus gland for T lymphocytes. So I'm not going to describe it in any more detail, but I hope that the message is clear that we are going to test all of the B cells that are being produced in this way, and they're still going to be in the bone marrow at the moment. Before they are allowed to leave the bone marrow, they will undergo a rigorous central tolerance process where we will test them against loads of self-peptides. So it involves the use of that protein, the autoimmune receptor, the AIRE. So I'll just remind you of that important protein. So it involves the use of this protein by certain cells in the bone marrow so that they can produce proteins from all over the body, and they will be testing the B cells that we are producing against all of these different proteins of the body and making sure that the B cells do not bind too strongly to those proteins of the body. We want to make sure that their B cell receptors cannot bind to these proteins too strongly so that the B cells won't be activated against self-peptides and won't cause an autoimmune response. Then, once the B cells have passed the central tolerance process, so the B cells that do not have a B cell receptor which can bind to self-peptides, these will then be allowed to leave the bone marrow and they'll go into the bloodstream, and once they're in the bloodstream they'll do exactly the same thing as T cells did. They will circulate between the bloodstream and also the lymphatic system. So they'll leave the bloodstream via high endothelial venules and go into lymph nodes, and then they'll move through the lymphatic system. So let me just get my pictures back up for this if I can find them. Here we go. So here are my pictures that I drew for the T cell recirculation. So what will happen is the bone marrow will put them into the bloodstream, and then they'll go around the bloodstream a couple of times before they eventually go through microvasculature of a lymph node, and then the high endothelial venules, these special types of venules in the microvasculature of lymph nodes, will recruit them from the circulatory system into the lymph node. And then once they're inside the lymph node, the B cells like to hang out in the B cell follicles. So they'll go to B cell follicles, but they won't remain in the B cell follicles forever. They, like the T cells, will pass from B cell follicles into the medullary sinus, and then they'll move to other lymph nodes, and they'll drain through the lymphatic system gradually until they drain into one of the great lymphatic vessels, so either the thoracic duct or the right lymphatic duct, which will return them into the circulatory system, and then they go around the process again. So the point is that B lymphocytes are continually circulating around the body and they're moving to different lymph nodes all over the body, just like the T cells, to make sure that they spend a little bit of time all over the place where we might actually need them. Okay, the final thing to say then is that their lifespan is similar to the lifespan of naive T cells. And by the way, we would call these B cells now we would call them mature naive B cells. So they are mature, their development process is finished, but they are currently naive because they have never seen their antigen yet. They've never been activated as part of an adaptive immune response. So the lifespan of a mature naive B cell is similar to the lifespan of a mature naive T cell. It's from months to years. So they'll circulate between the circulatory system and the lymphatic system for months to up to a year, or maybe even beyond that, and then they'll be removed from the bloodstream. The difference between B cell production and T cell production is that B cell production will occur throughout your lifetime. The bone marrow does not involute like the thymus gland involutes, so you can continue producing more and more B cells throughout your lifetime. So the process that I've described will continue on throughout your lifetime. Okay, so overall then, that is the production process for B cells now complete. What we will move on to in the next video is discussing the actual activation of B cells as part of the humoral adaptive immune response. The key concept that I hope you have taken at least from this production of B cells discussion is that we will produce a huge, great variety of different B cells, and indeed actually a concept that's important, which was important in the case of T cells, is the concept of a B cell clone. So this means exactly the same as we had with the T cell clones. It means the population of B cells with an identical immunoglobulin antigen binding region. So all of the B cells that have an identical immunoglobulin molecule as far as the antigen binding region is concerned, and therefore will be targeted against the same proteins. So the key message that I hope you've gained from this is that we will have a huge number of different B cell clones, and that these B cell clones will not be activatable by self-peptides. So we have destroyed hopefully all of the B cell clones that could be activated by self-peptides, and now when we have a pathogen infecting a portion of tissue, hopefully we will be able to activate certain B cell clones which will be able to recognize epitopes of their antigens. Okay, so we'll come on to the activation of B cells in the next video.